Good afternoon, everyone, and thank you very much for inviting me to be with you here today. My name is Dr. Rizig. I am a Senior Clinical Research Fellow at University College London, and I'm delighted to speak to you today about the subject of Parkinson's disease genetics, and I will be covering the lessons which we learned from the genetics of Parkinson's disease within the last 20 years. So as all of us are aware, Parkinson's disease is a progressive neurodegenerative disease. It is characterized by movement disorders involving motor skills and non-motor symptoms, and is mainly related to the accumulation of alpha-synuclein in the brain and other tissues around the body. So all of us are aware, Parkinson's disease figures are on the rise all around the world, and we are expecting by the year 2040 for these figures to be doubled. And as we are aware, Parkinson's disease is affecting all races and all cultures. This slide has been taken from the Global Burden of Disease study in 2016. And as we can see here clearly, that Parkinson's disease figures or Parkinson's disease cases are spreading around the world. And particularly, we noticed an increase in the figures around Africa and the Middle East. We're expecting these figures to reach about 40% rise in the year 2050. This particular slide is showing the last 200 years history of Parkinson's disease from the time when the disease was described by James Parkinson in the 1817. I like this particular slide because it demonstrated that within the last 20 years, from the 1997, the discoveries in the field exceeded all the discoveries in the first 100 years. And this is basically related to the genetic discoveries and advance within the genomics technologies. There is a great shift in the understanding of the etiology of Parkinson's disease within the last 20 years. So in the year 1996, we used to think about Parkinson's disease as predominantly caused by environmental risk factors. And through the development of the genetic technologies, we realized that the genetics is playing a significant role in the etiology of Parkinson's disease. And now we think almost the genetics are the predominant factor, which is contributing to the pathology of Parkinson's disease in comparison to the environmental factors. As I said, the advance in the genetics technologies made the discoveries within Parkinson's disease easier, certainly. And these discoveries started by linkage studies and GWASs and the ability to do functional studies and building databases and biobanks. So what we've been able to achieve is we discovered a lot of Mendelian genes related to Parkinson's disease. We've been able to identify risk loci and quantify heritability risk and polygenic risk scores. In addition, through these technologies, we've been able to build pathways and understand pathophysiologies and gene functions related to the disease. And certainly, we developed a lot of understanding for biological systems, and we've been able to integrate this into health informatics and artificial intelligence. And how this has been implicated in clinical practices, we've been able certainly to diagnose the disease better. We're hoping in the future we will be able to apply these into technologies related to genetic counseling and drug discoveries. And through this particular talk, I will speak about applicability of these genetic discoveries to predict the disease in different populations. And the ultimate goal is to use all these discoveries to enhance diagnosis, prognosis, predictions of outcome and screening of patients, and certainly to understand the phenotypic and the clinical variability of the disease. So to be able to understand the genetics of Parkinson's disease, we need to be able to understand this particular model of common versus rare allele hypothesis in complex disease. And by complex disease, we mean common diseases with a prevalence within a population of more than 2%. And this particular model identifies how rare variants and common variants can contribute to complex diseases like Parkinson's disease and other neurodegenerative conditions. So we can see here in the y-axis is the effect size, and in the x-axis is the allele frequency. The effect size means how deleterious the certain mutation could be. And we can see in the left corner of this particular slide that there are certain rare variants which are really rare within a population of allele frequencies less than 0.001. And some of them, they do have very high effect and deleterious effect if they get inherited. And those particular rare variants normally get inherited in a Mendelian fashion and run in families. In the opposite part of the spectrum, there are certain alleles or certain variants that they do have a higher allele frequencies. And by higher allele frequencies, we mean that they are And by higher allele frequencies, we mean they are more than 0.05% in terms of allele frequencies. So they are certainly common within the population, and they do have a lower deleterious effect. So they do not cause, if they get inherited, they do have a modest or low effect of causing the disease. But the problem, if you do have a larger number of those variants within a certain population, you ended up having a higher risk of, or certain individual within a population, you ended up having a higher possibility of developing the disease. So this is the same slide that is reflected to the genetic of Parkinson's disease. So there are certain variants which we know that they're really very, very rare in certain genes, and those genes normally inherited within families. Inheriting those particular rare variants can be very risky because they are deleterious and they get inherited in a Mendelian fashion. There are a group of variants within genes with minor or intermediate allele frequencies. So they are not common, but they are not rare. And those certainly we know that they can cause Parkinson's disease within families or the general population as idiopathic in idiopathic cases. So they are not inherited within families. And we know that those particular variants have higher frequencies in certain populations. And we will come into that when we discuss the monogenic types of inheritance in Parkinson's disease. We will take example from the LRRK2 G2019S. At the end of the spectrums, there are a group of variants within genes. Those particular variants are commoner within the general population, but inheriting some of them or a group of them can increase the risk of certain individuals' variants are particularly difficult to pick up, and you need large studies in a case-control fashion to understand and dissect those particular variants. So I will start by discussing the monogenic type of Parkinson's disease. It's caused by mutation in a dominantly or recessively or X-linked inherited genes. They are relatively rare types of Parkinson's disease, and they collectively count for about 30% of familial and 3 to 5% of sporadic cases. Going back to the same slide, just repeating what I've said before, there are variants in certain particular genes. Those variants are really very rare. They inherited most of the time in Mendelian fashion, and inheriting those particular variants is certainly deleterious and almost associated with developing Parkinson's disease, and they run in families. So the first genetic mutation which has been described and linked to Parkinson's disease have been described on the famous Iowa Parkinson kindred in the alpha-synuclein gene in the NIH in 1997. And we certainly found that not just only pathogenic variants can cause the disease in the alpha-synuclein gene, but even alpha-synuclein triplication and the number of copies of that particular gene, or increasing number of copies of that particular gene, is associated with the disease. So these are the group of genes which have been identified as disease associated genes, and it's inherited in an autosomal dominant fashion. And what I would like to show in this particular table, that the number of those particular mutations increased significantly, or the discovery between 1997 to 2016, and almost all those variants are inherited within families, and they are normally associated with type of Parkinson's disease with atypical features. And they are related, almost all the affected individuals are almost young, under the age of 50. The most famous genes, which all of us know about within this particular group, is the alpha-synuclein and the LRRK2, and we'll discuss this in the next slides. These are the group of variants which has been identified and autosomal recessive. So by autosomal recessive, I'm quite sure that all of you are familiar with this. You need to have one copy in mom and one copy in dad, and the affected individual will be homozygous for this particular mutation to develop the disease. And the number of those particular mutations increased significantly between 1998 and 2017. Almost most of these particular genes are inherited in people who are really, or presented in people who are really very young. So the disease normally presents itself in the teens years, and then almost associated with atypical features like psychiatric features, dystonias, and seizures and cognitive declines in patients. And the group of genes which are really well known in this particular group is PINK1, DG1, Parkin, and FBX07, among others. With the PD, within this particular group of gene of monogenic, we do have a high risk genes as well. So inheriting them does not really cause the disease, but increase the risk of developing the disease. And within this particular group is a famous, the mutations within a famous gene, the GBA. So in total, we have at least 11 confirmed loci for BD, for the monogenic group, 3 dominant, 7 recessive, and 1 X-linked. So what do we know about these genes? These particular genes, we understand more about their functions. So we understand they're almost by doing the cell work and understanding more about the pathways which are related to Parkinson's disease, we could link them almost to certain particular functions within certain particular pathways. The mitochondrial pathways are one of those particular intracellular mechanism which can unify most of the genetic mutations in Parkinson's disease. So this particular slide show that how all the pathways or those particular mutations in this particular gene can converge into two main pathways: the mitochondrial pathway, which is the mitophagy pathways, and the lysosomal pathways, which is the autophagy pathways. And we know that from our knowledge and the intracellular studies which we've done on those particular mechanisms, we know almost both pathways are converging within the intracellular mechanisms and causing the disease. So now we're almost certain that there's certain link between autophagy and mitophagy, and we call it almost the common pathway of Parkinsonism, or the degenerative mechanism which are related to Parkinson's disease. Another lessons which we want to pay attention to, and I've taken an example from the LRRK2 gene, is in each gene of which is being inherited in a Mendelian fashion, there are multiple variants which we know that they can cause the disease within families. For the LRRK2, we know there are about 300 different variants within that particular gene. 20 variants at least are deleterious or pathogenic. We know these particular variants, they do have different distribution across the world. We know that the G2019S is the commonest mutation and it's being described around different populations from around the world. There are certain ethnic variability on the way those particular variants within the LRRK2 gene are distributed around the world. This particular map is being taken from the MDS gene website and is showing that the distribution of the G2019S among different ethnic populations. So the majority of the G2019S carriers, as we know, are coming from an Ashkenazi Jewish background and in Northern African populations. And in Northern African populations, and there are certain areas around the world where the prevalence of the disease or the prevalence of the mutation is not known. I've just pointed few areas around the world where these particular mutations have never been described before, including Africa, Central Europe, Russia, and Central Asia. So this particular review is showing that the distribution of certain particular variants are really different across different populations. So if we look at the LRRK2 known variants, there's certainly 6 mutations which has been described consistently in different cohorts around the world. So the G2019S and the R1441CGH are the commonest by far and have been described in European populations. And as I said, in Ashkenazi Jewish population, particularly the G2019S and the North African. In the other end of this, certain mutation which are really common in populations within Asia, specifically East Asian and the Chinese populations, like the G2385R and the alanine to valine mutation in the 419 position. And so this particular slide is showing how particular variants within one gene or a group of variants within one gene can vary significantly among different populations. So the other lesson which we learn from studying the genetics of Parkinson's disease is we started actually translating this into promising clinical applications, such as biomarker studies and different clinical trials. So for the LRRK2 study, promising few studies looking at exomes in urine and CSF. And there are promising clinical trials with the LRRK2, including the Denali LRRK2 inhibitors. And there are similar studies relating to the GBA and mitochondrial functions and alpha-synuclein pathways related to Parkinson's disease. So a lot of those genetic discoveries have been translated now into biomarkers and meaningful therapeutic trials. In addition, we are building promising applications, including databases which are looking at both phenotypic and the genetic variabilities in Parkinson's disease. And these are really very useful as clinical tools for clinical practices in neurological services. So what we've learned so far. We live in a time when instead of having one gene for a syndrome, we're having multiple Mendelian and risk loci. Genetics could be used to refine the phenotypes of the disease, and genetic risk factors share common functions in intracellular pathways. We spoke about the lysosomal pathways and the mitochondrial functions, and understanding these intracellular pathways could aid our understanding of the neurodegenerative mechanism, variability in phenotypic feature, and help devise new treatments. Unfortunately, Mendelian loci explain only 10 to 20% of cases. So alpha-synuclein, for example, explain about only 10% of the disease risk, LRRK2 about 2%, GBA 5%, and other Mendelian genes about 2%. So these figures vary between different populations. So for example, the LRRK2 and the GBA are common in certain populations. Certainly the LRRK2 is very common in Ashkenazi Jews and North African. So how we could explain the remaining 80 to 85% of PD cases, and we call them idiopathic cases because we do not know what is causing these cases. So to identify the genetic risk or the genetic contribution, we have to use different methodology. And this methodology is using the disease model within the common variation in the general population. So we use the Genome-Wide association study, which is an approach that involves a screening marker across the complete genomes of many individuals to find the genetic variations related to specific disease. And these particular type of studies are really useful in finding genetic variation that contribute to common diseases. So this is the same slide as I presented before for the monogenic disease, and we can look at the end of the spectrum here where the common variants which are inherited in the population in a higher allele frequency or minor allele frequencies they are low risk. Sometimes they cannot cause the disease directly in family, but inheriting some of them or a group of them can increase the risk of certain individual to have the disease. So the number of GWAS, the GWAS started in Parkinson's disease in 2006, and then they increased dramatically. They started in Europeans, and the majority of those studies have been conducted within the last 10 years in European populations. And we can see in this slide that the numbers increased, number of patients, including these particular studies, increased significantly. So the first study started by 2,000 patients, and the latest one, which has been conducted about 2 years ago, it's increased to 37,000 patients. So we can see that these are the number of studies between 2006 till 2019, and we can see that the number of the loci which has been identified through genome-wide association studies increased significantly over the years, and we do have now number of loci or hits in genome-wide association study exceeding about 90 loci. So this is the first study which has been conducted in the European population, and we can see that we do have about 2 hits. This number increased significantly in the last Genome-Wide Association study in 2019, where the number of hits exceeded 90. So it's about 90 or 92 loci so far. And as I said, it's being conducted in a very large cohort of European patients, 37,000 patients and 1.4 million controls, and it's been published in the Lancet in 2019. At the same time, other efforts have been conducted around the world. This is a recent publication in an Asian population looking at the risk of PD in Asian and comparing these risk factors to Europeans. And we can see that large number of those particular, so the black dots in here, so the black genes, which are labeled in black, are the replicated loci from the European populations, and the red one are the novel loci, which has been just described in the Asian population. And we can see that large number of the loci within the Asian population or even the European population have been replicated in the Asian population, and additional 2 new loci have been described in the Asian population. This particular Genome-Wide Association study have been recently added to the Med archive. It hasn't been published in a peer-reviewed journal yet, and is showing that in about 800 cases and 690 controls from Latin American population, they have been able to replicate the alpha-synuclein mutation or the alpha-synuclein loci in Latinos. And this is a very, very encouraging, because we can see that by conducting GWAS studies among different populations, we have been able to replicate the finding from the original studies in the European population. And we're hoping that by increasing the sample size and looking at other populations other than the Europeans, we will be able to identify more loci and replicate the ones which we already know. So just the recap, if we look at from 1997, as I said, till now, the number of genes which have been identified as risk genes or pathogenic contributor to Parkinson's disease or disease-related genes increased significantly. And certainly after the introduction of the GWAS or after the GWAS era, those numbers, the numbers of genes increases significantly and almost duplicated. One particular application which has been which resulted from the GWAS study and is particularly useful is the polygenic risk score. And a polygenic risk score is an estimate of individual genetic liability to a trait or a disease, calculated according to the genotype profile and relevant genome-wide association study. Polygenic risk score is particularly useful because you can stratify people based on disease risk, based on their phenotype, or predict progression, or use them in biomarker studies and treatment options. And certainly we've been really very successful through the Genome-Wide Association study in Parkinson's disease to predict the polygenic risk score in Europeans, and hopefully by applying the same GWAS approach into other populations, we will be able to extend the utility of polygenic risk score to the other population. So to recap, for Mendelian PD, clearly we have been successful. Even for sporadic PD, we have come a long way. We understand about 30% of the disease risk. This is using both familial and the Genome-Wide Association studies approach. We are moving towards clinical implementation. So we know a little bit about the biomarkers, therapeutic targets. We build up databases. We're thinking about applicability of approaches, clinical approaches like reimplantation, genetic diagnosis, and we're thinking about disease stratification. But 90% of those findings, as I said, has been in Caucasian. So these are European populations. So certainly we could do better. And by mean doing better is extending our study to include people who are underrepresented currently in genetic studies and GWASs. And this has been something which has been shouted in the scientific community, been discussing a lot of publication within the last 2 to 3 years, because we are lacking diversity in our genetic studies and GWASs. And we can see here the majority of the GWAS studies in the GWAS catalog are coming from European populations. So why do we think this is really very important to study understudied population? Because there are significant genetic variability between different populations, and this is based on allele frequencies, linkage disequilibrium, certainly having different genetic admixture between different populations, beside the environmental risk factors and contributors to the disease. And when we think about individuals living within the Middle East regions or North Africa, there is a huge contribution from different population and migration across the history. And this certainly contributed to increasing linkage disequilibrium and genetic admixture within those particular populations. So what are the implications of these genetic differences in PD risk in different populations? I mean, certainly those particular genes which are contributing to Parkinson's disease in the European population could be replicated within the population from the Middle East and North Africa. But in addition, because of the differences within the genetic architecture of the population living in the MENA region, certainly there will be new genes which are contributing to Parkinson's disease risk. And we're expecting as well there are new variants within the known genes which have been described or discovered in the European population or the Asian populations. In addition, all genes which are contributing to Parkinson's disease can contribute to completely different diseases or neurodegenerative conditions within the MENA region. One important application of studying a disease risk within different populations is trying to identify the disease pathology better. And by identifying the disease pathology better, I mean comparing the genetic risk and the contribution between different populations through trans-ethnic mapping. Trans-ethnic mapping is a particular approach which can harvest and leverage the genetic diversity in one population, for example, African population, and combine it with another population like the European population, benefit from the linkage disequilibrium in one population to discover causal variants and rare variants in both populations. And this is a very powerful strategy to locate causal rare variants and common variants within different populations. So certainly this is something which the MENA region can benefit a lot from due to the high admixture within the populations living in that particular region. So, and even if we extend our approaches to include multiple population, we will be able to enhance on our understanding of the discoveries from the GWASs in the European. For example, we will be able to improve disease prediction by having a better polygenic risk scores from the European population. So if we include variation or variance which has been discovered in multiple ethnic population, we will be able to have a very strong, powerful polygenic risk score which is which can enhance the predictability of the disease, not only for the European, but across the ethnic groups and help with the disease stratification. Another benefit from involving other population in GWAS studies is we will be able to understand the disease better, because if we discover other variants which are related to known genes or new genes which are related to a certain disease, we will be able to map them on pathological mechanism. And what we're predicting is all these particular variants or genes will line within certain pathways, and these pathways will converge at some point. So we will have a better understanding for the intracellular mechanism and the pathways which are relating to the disease. So there are certain challenges. I wouldn't go into details about them. So when it comes into the Mie Rizig, for example, there are no cohorts which are coming from that particular region to be able to study the disease. There are certain technical difficulties related to genetic studies in underrepresented population. For example, there are no reference genomes, there is no imputation panel, there are no platforms which are suitable for the genotyping on those particular populations. There are certainly need for technical expertise to conduct genetic studies, and there are other issues related to cost and ethical consideration. So these are some solutions which I'm presenting here, and I hope that you will take home and start thinking about it. To resolve all these problems and enhance genetic discoveries within underrepresented populations, certainly within the Mie region, we need to think laterally, so we need to think outside the box. We require to work in teams, because genetic studies, especially GWAS studies, require large cohorts up to 1,000 or hundreds of thousands. We need collaborative approaches. Certainly we need to build databases, registries, and we need to use and harness technology. And certainly you need to have a lot of involvement from the patients and the public around you. So you encourage recruitment and enhance your cohorts. So the take-home thoughts is most familial studies in GWAS and PD have been in the European population. However, PD risk in two populations may be driven by the same or different underlying causal variants. So we need more studies to identify those variants. With the focus of genomic and personalized medicine, such European-only findings have very limited clinical utility. Thus, identifying shared genetic component will help us uncover causal variants and novel disease etiologies. So we need to identify families with PD in understudied populations, such as the population in the Mie, to help us find the genetic causes of Mendelian diseases in these populations. In addition, we need to examine PD mutations in Europeans, which has been discovered in the Europeans, and replicated them in understudied populations from the Mie region, for example. And we need to use GWAS approaches to localize ethnic-specific loci for complex PD and compare these loci to previously identified Europeans loci. So what are the predictions for the future? It's almost certainly we will find new variants that can cause and predispose underrepresented population to PD. We might find genes in similar pathways to the pathways which have been discovered in the Europeans, including the mitophagy and the autophagy pathways. And by applying this, we will get better understanding for the disease pathology and diagnose the disease better and manage PD in a better way, especially in underrepresented and ethnic diverse cohorts. And thank you very much.